So, good evening, everyone. For today's lesson, sorry for the time and the whole delay. So, for today's lesson, we'll be looking at the foundations of life. So, this is our week one biology lesson. And for this lesson, we'll be looking at the foundation of life. So, as usual, those that have been previously attending, they already know what I'm about to say. This lesson is a 12-week lesson where if you stay with us through this 12 week, you'll be able to learn your entire syllabus, covering all of them as much as you can, or having assistance in learning as much as we can throughout the entire syllabus, such that within 12 weeks, that's in 3 months' time, you'll be completely done with your semester, with your syllabus from 001 up until 004. So, in case maybe in your school they've not already been taking your lectures seriously, or maybe some lecturers have not already been paying so much attention to you guys, this is like an advantage to probably have someone teach you at least, even though it might be on a kind of speed, but on your level, it would help you if you want to study, you would know what you are looking at. So we'll try to be as simplifying as possible. We'll try to make sure the lesson is as simplified as possible, sorry. And yeah, so let's head into the lesson. So, foundations of life. So first off, in this lesson, we'll be looking at, by the end of the lesson, I mean, we'll be able to define biology and identify its core branches and the importance in modern society. So I'll be fast because we are kind of behind time. So, and we'll also try to see how we can apply scientific methods to biological experiments and analyze data using basic biostatistics. So also we'll also be able to describe the origin of organic molecules and the evolution of first cells. We'll also be able to explain the structure and function of key biological molecules like carbohydrates, lipids, proteins, nucleic acids, and their likes. So we'll also be able to master the cells theory and differentiate between the organization of plants and animals. I don't know whether it's organization or organization, but anyone, you understand what I'm saying. So first off, what is biology? What is biology? So biology is the scientific study of life. I don't know if you guys can still hear me.
Yes, sir. I can hear you, sir.
Okay. So biology is a scientific study of life and living organisms, including their structure, function, growth, evolution, and distribution. I don't know if this slide has been shared to you guys on the group. Probably you could use it to follow along. I don't know if it has. If you were in the lesson on Monday, I think the class was supposed to hold on Monday, but for some reasons it didn't because of I think network and issues with the tutor that was trying to teach you that Monday. So biology is the scientific study of life and living organisms, including their structure, function, growth, evolution, and distribution. So these words now that we mention in this definition, they are not just random words, or they're not just maybe we're not just trying to say things that maybe just to fill in the gaps so that the definition will be long. They are very important um, keywords that you would need to mention when probably if for any reason they ask you to define what biology is. It is the study of life and living organism. That's the first thing you should pay attention to. Sorry, the study of life and living organisms. So life, talking about the environment, like existence as a whole, and living organism, talking about what dwells in that particular realm of existence. So, and we're not just studying these living organisms, we are studying them and looking specifically at their structures, their functions, their growth, their evolution and distribution. So structure is not the same thing as function. Structure is talking about what they are made up of. What they are made up of. Do they have hands? Do they have legs? Do they have? Are they just moving on their structure? That's what they look like. Their outer appearance. Then their function, talking about what they do, whether what these their body parts do or what these organisms do in the environment. That's why we study the function. Also, we study the growth, how they grow. There are different phases of growth. That's where you see all those metamorphosis. That's where metamorphosis falls in. So, also looking at the evolution, how they've developed over time, how they've developed. Because evolution, when you talk about evolution, you're talking about their ancestry lineage or lineage, how they've flown from previous time or far back and how they've developed up until now for survival reasons. Then also their distribution, where we find them. That's where we come into habitats and their likes. So this alone, this particular definition helps you to know beforehand what the entire biology we are studying is going to be about. It's going to be about living organisms, it's going to be about life as ecosystem, the structure, the function, the growth, the evolution, and the distribution. So it seeks to understand the living machine at every level from DNA to ecosystem. So there are primary branches of biology. There are primary branches of biology. It's not that these are the only branches, but these are like the key branches that other ones could probably fall under as sub branches. So these key branches are botany, which is the study of plants, zoology, the study of animals, microbiology, the study of microscopic organisms, and genetics, which is the study of heredity and their variations. So sometimes some other texts you might see some other branches of biology, but these are the basic or the main, if I would put it that way, the main branches of biology: botany, zoology, microbiology, genetics. So this simple definition that we have here, they should be simplified enough to help you have a grasp of what they're talking about. Botany is the study of plants, their different features, their structure, their function, their growth, their evolution, their distribution. That's what botany is talking about. Zoology is talking about the same thing, the plant, but in this case, animal, the structure, the function, the growth, the evolution, and the distribution. Microorganisms, or that's microbiology, is doing the same thing, but now not to plant and animal that we can see, but to the ones we cannot see, doing the same study. Then genetics, doing the same study of the heredity and the variations, how these things have been changing over time, specifically trying to look at the evolutionary study. So that being said, let's see we have that how biology relates to our daily life. So there's something I found out. I was trying to create a content for someone, and it's a scientific content under biology, and I found out that basically almost everything seems to just fall under medicine, agriculture, and forensic. When you talk about biology, medicine, agriculture, and forensics, sometimes to enter biotechnology, but that's still under medicine. So biology, it spans into different areas of our daily life, but I'm going to be listing just these three because I found out that they seem to be recurring when we study forward. So in medicine, it helps us to understand human anatomy and the pathology. Sorry, it's the medicine. In medicine, biology helps us to understand human anatomy, and pathology allows us to develop vaccines and treat chronic diseases. So because of the study of biology, or because of the a branch or the use of biology, we can be able to understand the anatomy of humans. That's what we are made up of, what humans are made up of, not just animals, but now humans specifically, what they are made up of. And when we understand what they are made up of, and we also study some other things randomly around humans that influence humans' existence, we'll start finding out that okay, microorganisms sometimes they affect humans, and we can study these microorganisms, and from the study, we can be able to see how these microorganisms affect us adversely, then develop vaccines to fight these microorganisms depending on what they do to us. Like what happened during the COVID-19 period. There was that outbreak now. We didn't even know whether it was true or false, but whether it was true, whether it was false, I believe it was true. The virus was released, and for months, scientists continuously went through labs. I think I remembered my that period, my my lecturers, some of them that are advanced, are not just limited to Nigeria. They were telling us, they told, I think one of them, one lady, she told me about one of the experiments that they were carrying out, not in lab, but through what they call cheminformatics. It's a kind of programming form of chemistry, trying to predict drug or try to study drug structure and predict whether this particular drug will work for a particular disease. That was what they first did to discover the vaccine. They first tried it with programming. When it worked, or when it seemed like as if it was able to do the function, they now then try to see whether they can develop the vaccine rather. Because how most drugs work is that drugs, they would usually bind with the cause of the issue. Once they bonded with that cause of the issue, they can then prevent the thing from functioning. They will hinder the function, or they will start inhibiting the ability for that thing to function. So that's how they design it. But that's story for another day. So another way that biology relates to our daily life is agriculture. By the applying of genetics to crops and to crop improvements, it ensures that food security by creating pest resistance and high yield plants is possible. So I remember which some of us might be wondering before, they always say that famine happens. Like from the Bible, you said there was 7 years of famine during the time of Joseph. So how would there be famine for 7 full years? That is, and if you're someone that's really studies the Bible, you find out that this famine was not just something of maybe things hard. It was real scarcity of food. There was really no food that people would be willing to become slaves just to get food to survive. So this was how it was bad. So most of the time, the reason for famine sometimes is due to uncontrolled ecology or ecological systems, when probably plants come and start feeding on animals, like just eliminating these animals, like eliminating these plants. Plants are supposed that people will probably plant, and probably for all these kind of plants that maybe they will stay a year before they would harvest. Before it's the time for the plants to probably germinate, or sorry, not germinate, before it becomes time for the plants to be ripe, these pests, or what they call them, they would come and feed on all this crop in masses, especially locusts, in huge mass or in huge number. And before you know it, they've wiped off like huge number of food that should probably last for months. So if this kind of thing happens for a very long period of time, definitely the food supply will become low. And for some other reasons, this kind of thing happens. So, but due to the development of science and technology and even the study of biology, they've been able to create not just resistance to plants, not just pesticide that will kill the pest, but now they've engineered these plants to be able to resist this pest, or to be able to even repel the pest. Just imagine that normally rats usually eat crayfish. Then you now modify the crayfish such as when you make the crayfish to become crayfish, rats, it will repel rats. It will make rats run away from it. Rats will come, and once they perceive the smell, they will run away from it because of what you treated the crayfish with when it was alive. So that way, you've prepared what is going to be resistance to its own enemy. So that was biology was able to do, or was able to do through the study in the field of agriculture. So also we have in the field of forensic. So in this field, biology helps in fighting crimes, where you'd I think it happens. This one happens majorly abroad. It happens majorly abroad because that's where they really practice forensic science. Where you probably there'll be an incident, and probably they would be someone that they would suspect as the culprit, but due to examination of the body of the environment, they find out that okay, this person probably he was not around at that period. They didn't even find his fingerprint on the location, so they can be able to save the person and say, okay, oh, this person is free from, or maybe they would even check the murder weapon and find out that the fingerprint on the murder weapon is another person's fingerprint, and they'll be able to find who the real person is, and solution has happened. So this is how biology has been able to help in these different um, aspects of our human life. So now, this being said, sorry, this being said, there are several scientific methods that scientists have been able to use to carry out their biological experiments to cause the development we've been talking about. We talked about development in the field of medicine, talked about development in the field of agriculture, talked about development in the field of forensic. So how did they actually come about this whole development? How did they make up this study, or how did they carry out this study? You know, we said biology is the study of life and this. So how do they carry out this study? So first off, they would always start with the first phase, which is the observation phase. Observation phase. So what this observation phase talks about, or let me first list everything else out. I can probably use a story to explain it forward. So we have the observation phase, the questioning phase, the hypothesis phase, the experimentation phase, the analysis phase, and then the conclusion phase. So how does this thing all come together? So first off, how they do it, that they will first look at something that looks like something is this thing is, I don't really know what something is happening. Okay, let me use the incident that made people discover microorganisms. Before, they were always believing that any human being that fell sick was spiritual. Maybe spirits came and infested this person, probably the person started falling sick and the person died because they did not even know. So that's what they believed before. So then due to studies or some people that we are observing, they started observing that, okay, it seems like as if people that, there was one incident that I think they found out that people that sterilized their hand or washed their hand and treated patients, the patients in that their ward, they tend to, the death rate of those wards became less. Why those ones that don't treat their hand, the death rate was higher. So the scientist that was involved was like, he observed that difference. He was like, why is it that we felt that this thing was just spirits? Well, I'm observing that these same people that are sick, when people that wash their hand attend to them, many of them live. But in the other place, these people they don't wash their hand, and majority of the people that are sick, they always die. So they started observing something. They were like, something is wrong. They started asking questions. That was the next thing that started happening. They started asking, what is the reason for this? How is this thing linking to washing your hand? Is it that the water is what is making the people heal? They started thinking, when they started asking themselves question, they started suspecting, say, okay, maybe it is the water that's making people heal. And by making an assumption, you are doing what they call hypothesis. You are creating hypothesis. Hypothesis is like as if you are creating a supposed reason for it. You've not tested it, but you're saying maybe the reason is this. That an hypothesis is what you assume to be the possible cause. So the answer, okay, probably the reason is water. People that wash their hand with water. Maybe that's what usually heal people. Hypothesis one. Hypothesis say maybe there is something that they are washing off from their hand, or that the water removes from their hand that makes them, or maybe there's something on their hand that if they touch this person with this hand like that, the person will fall sick, or the sickness will become worse. That if you now wash it, the thing will go away. Hypothesis two, there is something on the hand that is causing it, not really the water that is causing it. It's not the water that is the solution. Maybe there's something that's on the hand. Then hypothesis three, maybe it is a spirit that is really causing it. Maybe these people are spiritual people. Maybe it's because of their, maybe it's because of their Igbo people, or maybe they are Igbo people, or maybe they are Yoruba people. That's why these people are being healed. So they say, okay, let's test this our hypothesis. So they now carry out experimentation. They say, okay, since we have three hypotheses that I just created now. I said the first one is maybe it is water that is the solution. Maybe we give these people, we will pour these people that are sick, we pour them water, maybe they will recover. Because if just washing your hand is making people live longer, maybe if we pour them the whole bath of water, maybe they will become healed. So that's experiment one. So they started doing experiments, pouring people in both places. They pour them water, pour them water. People were still dying in the same race we were dying. People that were still sick in the other place that they don't usually wash their hand were still falling sick. They were like, probably it's not the water. That hypothesis has been cancelled because they pour water on everybody, they bath them with water, but they still died. So that hypothesis was proven wrong through the experiment. Then the next thing is, or they carry out the experiment, then they now recorded, okay, did the death rate increase? That is the analysis. When they're checking, did the death rate increase? That is analysis phase, where you're checking whether did the death, as we pour this water, did they die more or did they die less? When they check, nothing changed. Nothing changed. That means it's not really the point of water. Then I say, okay, this washing of water, maybe something's on the hand. Oh yeah, let's wash their hand. Let other people wash their hand, and let's see whether if they even add soap, use something as an extra washing agent to wash their hand, maybe to even reduce the death rate more. They did that on both group of people, the people that are in the other in that other dirty side, and the people that are in the good side. They wash their hand probably because of there's something that is dirty on their hand that is causing the sickness, and they carry out the analysis. They saw that the death rate dropped. They said maybe it is because it is Igbo people, or maybe it's Yoruba people, or even American people. They said they will bring both American people and both Nigerian people, both Igbo people, put them on both places. Wash your hand. Some of them don't wash your hand. They tested them. They did the same thing. They tried the hypothesis. They found that it was not just whether it is American people. They found that it's not just pouring water. They found that, okay, something is actually on the hand that actually causes this sickness. So that way they've been able to come to a conclusion that there is something that if you don't wash your hand, it would cause sickness to get worse and cause more death rate. So this was how they started discovering cures, or started discovering some of the microorganisms that we know at the beginning. So they follow this procedure of first observing a particular thing, then next questioning why it happens, then carrying out hypothesis or making assumptions. Then next they experimented based on the assumptions. Okay, for assumption one, let's see whether assumption one is the real cause. They tried the experiment, they tried. They wrote the analysis down. They tried hypothesis two, assumption two, try, wrote the result down. Experiment three, try. That'll be assumption three, try, wrote the result down. Then they checked the result, they analyzed all the results. Then from the analysis of the results, Then I'm like, oh, this is the real solution here. We were able to draw a conclusion that it was not the first one, neither was it the third one, but it was the second method that is the real solution. So this is how scientists carry out experiments or carry out scientific practices everywhere. This can be applied in physics, can be applied in chemistry, can be applied in biology, can be applied in any field of science. They would always start with an observation, questioning, hypothesis, experimentation, analysis, then conclusion. In some other cases, they will, after conclusion, they will now say, okay, to make it a generalized something, whether it is the same thing everywhere, they will now try the same conclusion in several other locations around the world. If the thing is the same everywhere around the world, then they will make it a law that this is the truth, that this law holds true everywhere. So that is it, Sha. So some of the instruments that they use for these scientific um, biological experiments, some of them include microscopes, um, centrifuge, and their likes. We know these instruments. We know most of them. So microscope, we have a microscope here, and we are seeing the instruments, the parts of the microscope. I think this one is something that everybody should know. You just, I don't need to start explaining what it is per se. It's evident, and we have many other sources where we could get the purer explanation of what this thing is. But this is a typical microscope. It's a typical microscope, and we have the labels for everywhere. So we have labels for everywhere. I don't know if anyone has any question before I move to next slide. Does anyone have any question before we move forward?
No, sir.
Okay, okay. So let's move forward. So moving forward, we then have biostatistics. So when they carry out their experiments and they get to the phase of analysis, they carry out what we call biostatistics. Biostatistics, where they will carry out quantitative analysis, counting, using numbers to see how they can draw conclusions. Using numbers to see whether they can draw conclusions. And for different reasons, they even started using these numbers for different other purposes. And that's where the field of biostatistics came out, when they don't use the numbers for just analysis, but now you start using it for grouping and study other studies, wider studies of biological life. The field of biostatistics came up. So in this field of biostatistics, they use stuff like we see in normal statistics, mean, median, mode, to examine stuff to say, okay, oh, this place, the population of this thing is more here, population of this thing is more here, that kind of stuff. So it's just normal statistics. And the idea of biostatistics is that the use of statistics to try to analyze or aggregate biological phenomenons. So that's just by the way. So example, we can see here from our previous slide, we looked at here, we see insect types. We had different insect types. We had butterfly, housefly, soldier ants, and termites, and we saw their numbers at different sites. In the first site, we have 18 butterfly, 5 insects in the first site, 12 in this site. So when you do the statistics of everything, you find out that the place where there is more insects is in this region, in the site A. So you can draw any conclusion. You can draw, depending on what you want to find out. This data can help you get many insights about it. You can get the mean number of insects in the site. You can get several things, just several things. So this is just basic statistics using numbers in biology to get information or to get more clarity about the behavior or the happenings in a particular area. So we have the mean, median, mode, and we know what these things are already. So now moving forward, let's talk about organic molecules. Organic molecules. So from what I have here, I said the Miller-Urey's experiment. The Miller-Urey's experiment. So first of all, let me read what is here. It said life originated from organic precursors in the 1953. In 1953, Miller and Urey, they simulated the Earth's early atmosphere and applied lightning. Within days, amino acid, the building block of proteins, formed spontaneously. So this proved that chemical evolution was a possible start to life. So what is this jargon I'm explaining here, or what's this nonsense I'm trying to say? So previously, there was always this belief in scientists or in science that life always come from previously existing life. That life cannot come from nothing. They always had the belief that if living organism existed, they would have to have come from previously existing living organisms. But from these guys' experiments, from this Miller and Urey's experiment, they found out that without even any organism, they were able to create organisms to appear without any precursor organism. When you hear precursor, it's talking about previous parent, like maybe parent's organism. So this experiment, what they did is that they took an enclosed environment and they made it look like the what they call it, the Earth's atmosphere. They passed electricity because in the atmosphere we have gases like methane, ammonia, hydrogen, nitrogen. So these gases, they fill these gases in a bulb, like what we can see here. Then they sparked electricity, which should simulate lightning, because lightning does not happen only when humans are around. Whether there is human or there is no human, lightning will definitely happen. So they found out that when this thing happened, when they struck the lightning, within some days, they started discovering that some organic molecules, and when you hear of organic molecules, is living molecules, they began to form. Some living molecules began to form molecules like proteins, and protein is one of the strongest or the basic building blocks for living organisms. So they now found out that they were like, okay, wow, if this thing is possible, despite the fact we did not put any organism inside this place, it means life can come from nothing. The life can come from nothing. There's something like a chemical evolution where chemicals can lead to the formation of life. It might not lead to the formation of human beings, but it will lead to formation of probably some kind of microorganisms. So that's what this experiment talks about. So in case you see it in questions where you say, what's the idea behind the Miller-Urey's experiment? It was the experiment that tried to prove that life could come from non-organic sources. It could come from chemical sources. Life, organisms could evolve from chemical sources. That's just the idea of this experiment. So now, um, evolution and first cells with fossils. So here we're trying. The thing that the argument that time that made these people, this Miller and Urey, they carry out the experiment was that they were trying to find what was the first source. What was the first source of life? So they had believed that okay, life should not exist. They said there was previously a life. So they were not like, okay, what is that previous life that would have happened? So they're trying to now do experiments, whether it's possible. So then from that period, some studies came up where people started finding out some of the early life timelines. So they started discovering that there are something like the first cells that existed. So these first cells that existed were called the prokaryotic, anaerobic, and heterotrophic organisms. So these organisms, they tend to they were noticed to have existed over 3.5 billion years ago. And I don't even know how this usually measure these years. They do say carbon dating, but it does not even make any sense. That's older than our own Earth. 3.5 billion years ago. That's very, very, very long. I don't think our Earth is as long as that. So they discovered that the early life organism, the early living organisms, they were prokaryotics. They were prokaryotic, and they did not have many cells. They were simple celled organisms. They discovered that also they were anaerobic. They did not really need oxygen per se to survive, and they they sometimes they fed on something else that is not really themselves. They could feed on anything. So these they are finding we are like what they started discovering. That, okay, the first organisms were having these characteristics. So they began to divide based on what they found. They started dividing studying forward, seeing how these organisms they evolved moving forward, how they evolved, and they started trying to see whether they could characterize these organisms based on a standard characterization throughout. So this characterization was what now led to the formation of these kingdoms that we are here. Kingdom this, kingdom that, kingdom this, kingdom that. They were trying to group these organisms because they were like, these organisms now, we found out that okay, some of them have specific characteristics that are similar, while some seem to deviate, while some also seem to be similar, like as though they were a mixture of both traits. So they started grouping this all the organisms based on a broad grouping. And this grouping, the first, or let's say the topmost grouping, or the broadest, yeah, the broadest, that's the word. The broadest grouping was based on kingdoms. So they group them based on 5 kingdoms. The first one being Kingdom Monera, the next one being Kingdom Protista, the next Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. So for the Kingdom Monera, they included organisms like bacteria that were less complex organisms, the prokaryotic organisms. Then Kingdom Protista are the one that are unicellular organisms, the eukaryotes, the single cell. Sorry, prokaryotes are the ones that are multi-celled organisms. Then the unicellular, the eukaryotes, single-celled organisms. While they also had groupings, fungi. These ones they were kind of different in behavior. They did not behave like the unicellular organisms. They did not really behave usual, so they gave them a different group. They behaved like as though they were dead, or they fed on stuff that were dead. So example of these ones are they call these kind of organisms fungi, or this group of organisms, the kingdom organisms on that kingdom fungi, and mushroom is among the kind of organism that falls under that. Another one we have is the kingdom plantae, which is the common plants that we have. Almost all the plants we know fall under. Then we have the kingdom animalia, which is where animals and even humans fall under. So these 5 kingdoms, they are very, very important kingdoms, and I think they are like the broadest grouping of all living organisms. So we can see here, we see some of the bacteria here, we see some other more. Then we are also seeing some other ones that look dead. Then the ones that are green plants, the kingdom plantae. Then we have animals, both fowl, chicken, all of them. So biomolecules. So now we have I remember when we were talking about that when that Miller-Urey's experiment was being carried out, they discovered the key element that is responsible for living organisms, and that element is usually protein, or would I call them mixture of both proteins and carbo and hydrogen, like hydrocarbons, yeah, organic molecules. They are the building blocks for life. They are the building blocks for life. So these kinds of molecules that they found to be the building blocks of life. In chemistry, they will call them organic molecules, but in biology, they specifically grouped some of them and they call them biomolecules. And some of these biomolecules, we have the proteins, the carbohydrates, the lipids, and some other ones. So for the carbohydrates, they found out that these carbohydrates there, or these carbohydrates, let me just talk about their characteristics. That's what they are. The characteristics of these carbohydrates is that they are used for energy storage by both plants and animals. They are used for energy storage. Sorry. So the main function of carbohydrates in humans and animals and even plants is to store energy. Is to store energy. Carbohydrates is basically to store energy, and they store this energy in form of starch for plants and glycogen in form of and in form of glycogen for animals. So if you study them, the digestive system, or even the liver, you will find out that that in the study of the liver, they will talk about how glucose is converted to glycogen to store it, and glycogen is converted back to glucose when there's need for glucose. So when there's need for energy rather. So carbohydrate is used for as a means for energy storage, specifically energy storage. While lipids, on the other hand, their function includes insulation, that is as a form of coating, signaling, and long-term energy storage. So carbohydrates are for short-term energy storage or for just basic energy storage, but lipids, they are used for storing energy for a much longer time, like very, very long time. Because examples of this liquid, we have the fats, we have the oils, and yeah, fat and oil basically they are lipids, but typically it's fats because oils are not. Sometimes some textbook will not group oils as lipids, but they are under lipids. So fats and oils. So they are used for insulation, they are used for signaling, that's to send signals to different parts of the body, and they are used for what they call it. They are used for um, energy storage, long-term energy storage. So in this signaling, they are the ones that are they contain fatty acids and some other stuffs. So these fatty acids they are the one we know as carboxylic acids. Carboxylic acids. So that's fatty acid. And if it is how you would react or carry out this or cause these fatty acids to react, and they would release what we know as esters. And if there is one key function, if you're a chemistry person that you've been observing chemistry, there is one key function about esters. And the thing is that, oh, not really key function, there's one key characteristic of esters, and the key characteristic is that they have smell. They are responsible for smell. They are aromatic hydrocarbons. So these esters, fatty acids, are used to create, and they undergo reactions that create esters, and that is how signals are being sent. So that's how they are used in signaling. So that's why I explained this, because it might be like signaling. How is it possible? Fats signals. So how it happens is that these fats, they undergo some kind of reactions in the body when they attach them to glycerol. So when they undergo this reaction, they will release esters, and these esters, when they are being released, they are aromatic, they have scent. So this from this smell, depending on the kind of fatty acid, and depending on the animal, and depending on what these animals interpret this smell for. Like if you notice, sometimes if you've killed ants, some kind of ant, there is this offensive smell that comes out. Sometimes that smell um a signal to other ants that there is danger in this zone. Other ants would stay away from that particular zone. So that is how they are used for signaling. So sometimes they are used to create pheromones, but that's for upper levels. So another of these biomolecule is the protein. So these proteins, they are polymers of amino acids. So these were the ones that we talked about during the explanation for the Miller-Urey's experiment, that the experiment was able to create amino acids, and these amino acids were like this, had like the starting blocks for proteins. So proteins, they are polymers of amino acids, and they are known as the workhorse, or the workhorses of cells. They serve the function of they serve as enzymes and structural support. So these are the functions of protein. So we've talked about the functions of carbohydrates. We said they are basically for energy storage, and we can say for energy storage for short term. But for the one before this thing, we said lipids. For lipids, their key function is long-term energy storage, signaling, and also what they call it, insulation. Yeah, insulation. So for proteins, on the other hand, their own functions are, they serve as the workhorses for cells. That is like energy source. That's a form of energy source for cells. For cells. For cells. So they are the workhorses of cells, and repeating of cells. So you would clearly understand that it's cells we're talking specifically about. So they also serve as enzymes that they speed up reactions in the body. Proteins, they speed up reactions in the body. So they are helpful to the body because they can speed up metabolic process or catabolic process, or they speed up processes or reactions in living organisms generally. So also they serve as structural supports and transporters. So they are the ones that are used to transport different things within the body, or across the body, or across the living organism's structure. So one of the key feature in proteins is the presence of peptide bonds. Peptide bonds. So this is what is responsible for the long linkage of these amino acids to form proteins. So these peptide bonds are what we see here. This thing that is circled in this yellow here. So I'm talking about here, this point here. So this yellow stuff is the peptide bond. So it is what links one amino acid to another amino acid. So because of the ability to form these peptide bonds, we are able to see amino acids form long chain protein. So from what I have here, I said peptide bonds, they are formed through the dehydration synthesis, or dehydration synthesis reaction between carboxyl groups and amino groups. So this is the carboxyl group here, COOH, and we have the amino group NH2 here. So when these two, when oxygen and hydrogen here bonds with, or removes, when they are being removed, oxygen, hydrogen, and hydrogen, when they are being removed, this bond forms. When oxygen and two hydrogen is removed, that is water, that is dehydration. So when there is a dehydration of this carboxyl, of this of two amino acids, it leads to the formation of these proteins. So that is how proteins are being formed, from the dehydration of amino acids. from the addition of amino acids, and they function as one of the key strengths of cells. So that's by the way. So these proteins now, they are also the ones that are responsible for or they are also these amino acids. I mean, they are also the structure also for nucleic acids. That's the DNA and RNA. So this DNA and RNA, they are responsible for this the storage of information and the transfer of information from different between organisms, parents and daughter organisms. So this nucleic acid is another example of what they call it of biomolecule. So we mentioned three previously. We mentioned lipids as first. No, we mentioned them. It was it lipids we mentioned first. No, we mentioned carbohydrates as the first biomolecule. We then mentioned lipids or fat as the second biomolecule. Then we mentioned proteins as the next biomolecule. Then now we are mentioning nucleic acids. So nucleic acids, they are the next biomolecule. And I'm mentioning the functions of each of these biomolecules. So for nucleic acids, they are of two types, the DNA and the RNA. The DNA representing the deoxyribonucleic acid, while the RNA representing just the ribonucleic acid. So the deoxyribonucleic acid is one that has been where oxygen has been removed from it, and its key function is for storage of information. Storage of information. We have it here. Key function of that is for storage of information. DNAs are mostly responsible for storing informations, while RNAs they are responsible for transfer of information from parents to from parent to daughter. So they are composed of what's known as nucleotides. They are composed of what is formed by nucleotides. So how they are formed, their own is they are still long chain structures, but now they are linked together. They are linked. Their linking bond is called the phosphodiesters. The phosphodiesters. So these RNAs, they are of many types. They are many types. We have the adenine, the guanine. We have some other ones, the cytokinins and the thymines and the all of them. They are under the amine groups. So they are the members of the nucleic acid biomolecules. So these are biomolecules. We mentioned four of them, and we mentioned their functions, and some of them we talked about their structure, how they are made up of. We talked about I think the one before this slide was what was that self proteins. We said they are formed through formation of that bond, peptide bonds, while this one they are formed from through the phosphodiester bonds. So now we are moving to cell theory. We're moving to cell theory. Cell theory. So this cell theory was actually part of the theories that people believed were how cells function. They believe that they are the guide to understand cell. So we have the first one. We say that every living organism is made up of one or more cell. Every living organism is made up of one or more cell, and I think this is still true. Every living organism is made up of one or more cell. They are either unicellular or multicellular, so long as they are organisms. The organisms are always either unicellular or multicellular. We cannot see a non-cellular organism. Or as of now, I don't think I have seen anywhere where they say there's a non-cellular organism. So living organisms are always unicellular or multicellular. That is the first principle from the cell theory. So the next one is that cells are the fundamental units of structure and function. So they are the fundamental units of organisms for structure and function. Some places you see that they are the structural and functional units of life. Some places say structural and functional units of living organisms. So just the main idea is that they are the key units responsible for structure and function in living organisms. Structure talking about how they look, and function talking about what they do of living organisms or what they can do, what living organisms can do. So the next theory is that new cells are produced from existing cells via division. So this is the next principle of the cell theory, that all cells exist from previously existing cells. All cells exist from previously existing cells, and they exist by a process known as cell division. through a process known as cell division. So now, as I was saying, we have different organizations of living organisms. When I was saying that we have the unicellular, multicellular, or colonial. I did not mention colonial because colonial is an example of multicellular. When you talk about a colony, when you're talking about many, so it's multicellular, the broad grouping is multicellular. So for unicellular organisms, these are organisms that exist as a single cell. One cell performs all the functions of life. Examples of this kind of organisms are the amoeba and the bacteria. I think it would be wise for us to probably go back and look at more of these examples. Unicellular organisms, amoeba and bacteria. So they are unicellular because they are made up of just a single cell, and this single cell is responsible for every single thing. Just like as though they have one body part, and that one body part is what all they need. The one body part is their mouth, is their leg, is their hand, is their this. So, but in this case now, it's talking about they have one cell that is responsible for every single activity that they carry out. So then the next one is colonial. So this is a group of cells attached together but function independently. They are a group of cells that are attached together but function independently. So they are kind of multicellular, but not really multicellular per se because they still are functioning independently. So that they call them that they function as colonies. So they might be multicellular groups together, or they might be unicellular groups together, but they're just group of cells that function together. So now the multicellular are the ones that specialized cells organized into tissue, organs, and systems. So for this multicellular, we have several cells, and these several cells they now have different roles that they carry out. So maybe you might have, let's assume the body or that particular organism has like 100 cells. Maybe 20 will be responsible for respiration, 30 will be responsible for reproduction, 10 will be responsible for another thing. So there are several groups of cells that have a particular trained trait. They are specialized for a particular function. They are specialized for a particular function. So that are multicellular cells. They are distinguished. They are a group of cells that are specialized for a single functions. For a single function, sorry, where you have several of different groups of cells that carry out in specific functions. Specific functions. So examples of these kinds of multicellular systems is our heart. Our heart. Our heart carries the function of what they call it of circulating blood. So everything that makes up our heart is combination of both cells and tissues. So when you move from cell, you move up to tissues. A group of cell form what we know as a tissue. A group of tissue form what we know as organs. And a group of organs form what we know as systems. So I'll be repeating this thing I just said. A group of cell that carry out a single function, they form what we know as tissues. Group of cell, same function, tissue. Group of cells, same function is a tissue. Then group of tissue that carry out the same function they lead to the formation of what we know as organ. So when we have several tissues that come together, they form the organ for circulation. That's our heart. Several cells came together having individual functions, and these several cells that they came together, they form individual tissues that make up the tissue that plenty tissues. These plenty cells came together in different groupings and they formed several tissues. Then these several tissues then came together as well to form cells form organs to form organs. So when you then have several organs carrying out a single task, you then have what you see as a system. So we have the circulatory system, which is a combination of the heart, the veins, the arteries. So all these things make up the circulatory system. It's not only the heart that is the circulatory system. So long as you're talking about the system, it's not only the heart. You're not talking about only one organ. You're talking about several organs. And these organs, we have the veins, the arteries, the heart, and so on. So those are the organs. But now these organs now, each of these organs now, let's just focus on organ. Organs, the heart alone is made up of different tissues. Different tissues. They are made up of different tissues. Muscular tissues, different tissues. different tissues. The heart is made up of different tissues. So that's for tissues. So now, when you now zoom deeper into these tissues, each of these tissues are made up of several cells. Several cells. And that is how the organization goes, from cell coming up to tissue, coming up to organs, coming up to system, then coming up. If you group an entire system together, if you gather plenty systems together, you will then form an organism. When you have several systems coming together, you will form an organism. So that's why we humans, we are living organisms, because we are a combination of several systems. We have our reproductive system, we have our circulatory system, we have our respiratory systems. So all these systems come together and make up the single organisms, which is human. So that is for that. So that being said, I was saying that there are also there is what we call the prokaryotics and the eukaryotic cells. So these two cells, they're supposed to be called a specific kind of cell, but now for some reason they don't just group them. They are kind of classification of cells, where some cells are classified as prokaryotes and some are classified as eukaryotes. So for the prokaryotes, this classification was done based on the features that they have. So these features that they have that is responsible for the classification is the nucleus, the organelles, the DNA, and the ribosomes. So for the prokaryotes, they don't have nucleus. They have no nucleus. They have no nucleus. That's for the prokaryotes. So also they have no membrane organelles. They have no membrane bound organelles. So they have their operation, their DNA operation is circular, and their ribosome is small. They have small ribosomes. So these are the characteristics of prokaryotes. While for eukaryotes, they have membrane. They have this thing, nucleus. They have nucleus, and this nucleus is membrane bound. So they also have organelles like mitochondria, Golgi bodies, the endoplasmic reticulum, and so many other ones. So these when you hear organelles are something that is smaller than cell, something that is smaller than cell. The several things that make up a cell are known as organelles. Several things that makes up a cell are known as organelles. You remember when you're having the diagram of cell, plant cell and animal cell, all those many small things that they say is inside cell, mitochondria, endoplasmic reticulum, and all those their likes, they are what are known as the organelles. So organelles come together to form cells. Cells come together to form tissues. Tissues come together to form organs. Organs come together to form systems. Systems come together to form organisms. So that's the flow. So for eukaryotic organisms or for eukaryotic cells, they are known to have nucleus. So nucleus is a specific organelle inside. So I can say this is like the key difference between prokaryotes and eukaryotes. They have nucleus, they don't have nucleus. So the every thing is just kind of a kind of iteration. Just like as if they're saying for prokaryotes, they say, okay, well, they have organelles, but their organelles don't usually wear clothes. They are not restricted. They're not restricted. They don't have membrane. They are not membrane bound. While for eukaryotes, they have organelles, and these organelles they are membrane bound. They are kept together by a particular membrane. So these are the differences between these kinds of cell. So normally they're supposed to say that they're supposed to call them either it's just a kind of classification. There are several classifications. You can classify them based on the nucleus. And when you're talking about classification based on nucleus, you're talking about the prokaryotes and the eukaryotes. When you're talking about classification of cells based on what they call it, the number of them, their number. You're talking about the what they call it, the unicellular, and okay, that's classification of organisms. I think I mentioned one other classification. Okay, I was thinking it was a classification. So, so that is for the classification of cells. We have two major types: the ones that are nucleus present, and the ones that are nucleus absent. The eukaryotes and the prokaryotes. So now, um, we'll be doing another you in this lesson before we run. I think we're almost done. Yeah, we'll be doing a kind of comparison between plants and animals, or plant and animal cells. So for plant cells, I think this thing is something that we are used to. For those of us that studied them, that have looked at biology, when we're talking about cells, plant cells, they are known to have chloroplasts, and this chloroplast is also responsible for photosynthesis in these plants. They are known also to have cellulose cell wall plants. Then also plants, they are known to have large central vacuole. Then also the plants, they are known not to have centrioles. So centrioles are only present in animals. Centrioles are only present in animals. Centrioles are only present in animals, but absent in plants. Large vacuoles, plants. Chloroplasts, plants. Cell wall, plants. So these are like the key differences between the plant and animal cells. The plant cells, they have chloroplast. It's supposed to make sense. Chlorophyll is what is something that is in green plants specifically. And this chloroplast is also responsible for photosynthesis. So chloroplast is only plant that undergoes photosynthesis. So that should stick in your mind. So cell wall, cellulose cell wall, that's actually the full name. Cellulose cell wall. So cellulose is the way that plants store energy. They store energy, cellulose. Humans store their own as starch. So plants, humans store their own as something different, carbohydrates. So while this thing, they store their own as glycogen, yeah, glycogen. While this one they store their own as cellulose or starch. So when this one they have this cellulose cell wall, that's a wall that guides their cell. You should have seen the diagram. Cellulose cell wall, rigid cell wall, and it's only seen in plant cells, while animal cells they don't have it. So these are like the key differences between these plant and animal cells. So this I would say this draws the curtain for our evening's lesson. I don't know if anyone has any question before we call it a wrap. Any question before we call it a wrap?
Okay.
Okay, I would want to ask a question without flipping, without flipping, or if you have your book there with you, you can peep into your book as if you are using a book. What is the name of the experiment that proved that living organisms could come from chemical sources? What was the name of the experiment?
Muller experiment, sir.
Miller-Urey experiment.
Experiment.
What's the name of this? Okay, very interesting. Thank you. Thank you. Thank you. Are you on the group?
Yes, sir.
Okay. When drop the number you would prefer a 500 naira at a time on
Thanks so much, sir.
for the answer to that. Yeah, you're
Thanks.
welcome. Yep. You're welcome. Should I ask another question? Should I ask another question? Okay, clearly who can clearly list the steps that are involved in scientific method as we had it today?
Yeah,
You can
the
pick from
first
your
one
book
you,
or
the first one you mentioned is about the observation that scientists will test how
no,
to
string,
huh?
please. For you, I don't know. I know you are doing a very good job to be listening, but I don't know if because I want to give another bonus. So if anybody else can answer the question before I count 1 to 10, then maybe you'll be given an opportunity. If nobody answers before I count 1 to 10, then you'll be given the opportunity to try it again. So just hold on first, so that maybe another person can get the bonus.
Sorry,
10.
sir. Question, sir.
Yeah, yeah, I can hear you.
Please, sir, come again with the question,
I
sir.
didn't.
The
Okay,
question.
I asked the steps that I listed in the lesson today. The steps that I listed in the lesson today that I said are responsible for that are the scientific method steps, as I listed them today. If you wrote
Okay,
it in
sir.
your book, so
Okay. Number one is the observation. After
mhm.
we have hypothesis.
Okay,
In after hypothesis, we have experiments. Then we have analysis, then conclusion.
okay. You said number one is the
Observation.
observation.
Yes.
Number two
Number
is the hypothesis.
then number three is our experiments. Number four, analysis, and five is the conclusion, sir.
Experimentation.
Yes.
So, are you looking at your book or you are not looking at your book?
I'm not looking at my book because I'm not with my book, sir.
Okay, you're not looking at the book. Okay,
Yes,
because of you attempted, you drop your number as well. I think, yeah, drop your number as well. Adeni,
Yes, sir. I'll
Abdul.
be calling
Yeah,
you.
drop your name in the group as well. You're on the group, right?
Yes, sir.
Yeah, Abdul you. So drop your number on the group as well. You want
Okay, sir.
yourself also a 500 naira at a time. So see you guys
The first
in our
question.
next meeting. Good night, everyone.
Okay.
Though your answer is not necessarily correct. You missed one thing in the answer. You missed one of them. But I'm giving you that because of the attempt. It's first you have the observation, then you omitted the questioning based on what we have on our slide. So there's a questioning phase. If you should look at it again, there's a questioning phase before you have your other stuffs. So, but because you attempted it and you were listening, so that's it. There are 6 steps, but you mentioned 5. So questioning comes before hypothesis. So well done, and see you guys when next we see. Good night, everybody.